Hello, hello, hello. I am. Ah, jeez, Louise. Okay, coolio. Where are we? Right. Let's make sure this is all connected. We are in. All right. Hi, guys. This is really an A-level live. And this is really for people taking their paper 3 exam on Wednesday, and it's just really to go through some beyond the specification information that you guys might be needing. Ah, right. Okay, let me move myself up. Oh my god, why's my head like a giant flipping? My camera's gone a bit big. Never mind. These things happen. Okay, let me just sort out this. So this is all in the right way. So I'm really just going to go through some extracurricular or beyond the specification information that you need to get the top marks on the essay, and then just show you where you can apply it. Okay, yeah, no problem, midget. That's all good. All right, so let me get into this. Okay, so when you do your biology essay worth 25 marks, you should spend about 45 minutes doing it. If you want to get the 21 to 25 marks, so the 4 bonus marks, like super extra marks for going beyond your specification, I'm going to go through what you should talk about to do with cholera. Cholera is something that used to be on the old specification, which ran up to like 2015. And in that specification, cholera was part of the AS or Year 12 content for biology A level. And it's a really cool bit of extracurricular or beyond the specification information, because this you can include in like so many different essay titles. So the binomial name for cholera is Vibrio cholera, as you can see here. Let me just get the old pen out. So this is the binomial name. So of course, this is your genus, Vibrio, and this is your species. Now, I wouldn't necessarily use this one in terms of classification. There's not an awful lot to go into about it. But if you were at a stretch and had nothing else to put down, you got the genus and species here. Of the cholera. Now, the structure of cholera is the same as all your prokaryotes. So if you wanted to waffle a little bit about the structure of cholera, it's the same as all prokaryotes and other bacteria. So what do you know about the structure of bacteria? Yeah, you did this in GCSE. That's right. Betty, I reckon it would be good, Vibrio cholera, for your paper 3. Like, it shows it's beyond the specification, so it's worth remembering. It's quite easy to remember as well, because the species name is cholera, and you've just got to add Vibrio. And what can trigger you to remember that it's called Vibrio? Well, the bit about the cholera that you've got to know is that it has a corkscrew tail. Now, do you remember the name of the tail on bacteria begins with an F? It helps them swim. Yeah, flagellum, nice one. Yeah, so the flagellum, it helps them swim. But more importantly than that, that's right, Betty, the corkscrew tail is gonna, and is gonna make, when that goes, it gives the cholera a kind of spinning motion, like hence the name Vibrio, like vibrating. Kind of, it's going to spin like that in a corkscrew fashion. So you really want to know that the flagellum creates a corkscrew motion. What that means is it allows the cholera to burrow through your intestines lining, the mucus layer that's on the intestines. Now, the mucus layer normally traps bacteria, as you know, here with your ciliated epithelial cells and stuff. You trap pathogens, and that's part of the defense against invasion from pathogens. However, cholera has an adaptation, which is this corkscrew flagellum. So yeah, Betty, if you can remember that it has a corkscrew flagellum that's going to allow it to penetrate the epithelial cells of your small intestine. These are the extracurricular things. So yes, the name, but also its flagellum is not beyond your spec, but the corkscrew motion and the corkscrew shape of the flagellum is beyond your spec. So what does it do? Well, it corkscrews through the mucus lining. So you've got a little bit of mucus here on your epithelial cells. There's goblet cells that produce this mucus to trap bacteria from penetrating your epithelium. It's got the adaptation, the corkscrew, to go through that. Now, once this bacteria is inside your epithelial cells, so once it's inside those epithelial cells, what does it do? Do you know anything? You may have seen some other people covering this for the extracurricular or beyond the spec stuff. Betty, do you know what it does? How it makes you sick? So once it's into your epithelial cells. Nice. Yeah. So actually, close. So, and this is going to be some information that will fit into two parts. So how it makes us sick? Well, point one, cholera produces two proteins and the first one is going to be one of them is going to do what you just said and is going to open those chloride ion channels. But before that, because this is going to work into some of the essays, one of the proteins that it produces two proteins. Remember, we're thinking about specific shape, 3D structure of the protein. The first one binds to carbohydrate receptors. So specific carbohydrate receptors. So I'm going to use the word specific. Carbohydrate specific receptors only found on epithelial cells in the small intestine. So it's only found there. So they're very specific, and this is why it doesn't infect other parts of the body, and cholera will only infect the small intestines. So this is going to be key when we look at where we can use this information and why we can use it in so many essays. Right. The more important bit is the second protein binds to your right chloride ion channels, opening them and allowing chloride ions, which you can use the symbol Cl minus, is an accepted symbol for chloride ions, and allowing chloride ions to move into the lumen. Now, why is that a bad thing? We're going to show why that's a bad thing. So, if I have enough space. So the chloride ions are going to now move into the lumen. We got these chloride ion channels open, and just to get the direction right, the chloride ions are going this way into the lumen where all your digested food is. So they're going to go in here, and that is going to change the water potential of your epithelial cells. So in your epithelial cells, what is going to happen to... I should have left myself a little bit more room here, shouldn't I? What's going to happen to the water potential inside the epithelial cells? And you can kind of follow this down its natural progression. So what will happen to the... If you take ions out of a cell, what happens to the water potential? The water is going to move into the lumen. That's absolutely right. But why is the water going to move into the lumen? What's happened to the water potential? Decreasing the water potential. Right. So first of all, the chloride ions leaving means there's less ions in the solution or in the cytoplasm of the epithelial cells. So because there's less ions in there, the water potential increases in the epithelial cells. So water potential increases in epithelial cells, okay, because the chloride ions have moved into the lumen. So what we really want to say is, here is number 3 in my list that's running out of room. Water potential increases in the epithelial cells. Now, what does that mean? That means water is going to move into those epithelial cells. Oh, sorry. Yeah, water potential increases in epithelial cells. So water moves into lumen by osmosis. And that causes... Yikes. That doesn't cause yikes. Water moves into the lumen by osmosis. And then we can go for 4 over here. And we can draw water leaving. Yeah, doing good, doing good. So water moves in over here, H2O, and that's going to cause diarrhea. Oh my days. Spelling diarrhea is almost as bad as having diarrhea. So that causes diarrhea and dehydration. Okay, so there's quite a lot of information there, but there's even more information. No, you don't need to know the names of the specific proteins at all. You've got enough information there to go on, really. That's a decent explanation. But why have I drawn the blood and why have I drawn the body cells? Well, we get a bit of an issue. A concentration gradient is set up between the blood and the epithelial cells, diffusing more ions into the epithelial cells. So we get a diffusion gradient, so we can bring diffusion into this, and then we can bring osmosis back into it by saying the water... no problem, Galland. And we can bring osmosis back into it because the water then moves from the body cells into the blood, into the epithelial cells, into the lumen. So what we're going to do is set up a diffusion gradient of ions going ping, 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 ping. And then we're going to follow that with a water gradient going ping, 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 ping. So it's not like it's just a bit of water moves in and it's all over. So 6 ions now diffuse into epithelial cells from blood and body cells down a diffusion gradient, like a chemical diffusion gradient. So they're not moving, they're not doing osmosis, they're moving down a diffusion gradient, and then of course that is going to set up the osmosis to follow down a water potential gradient, because as the ions move in, they'll lower the water potential again, and that will set the osmosis going from the body cells to the blood, etc. Then water follows by osmosis down a water potential gradient. So that bit's a little bit more complicated. Would you have to include that? No, but you could do. It's just the natural follow-on from that. I think I don't even know if I can fit all of this in. But so what am I actually on about there? Well, what I'm saying is at 6.6 over here, chloride ions and ions start moving in here, because remember we moved the chloride ions out, so now they're trying to move in here and move in here, and then 0.7 is that water is going to swiftly follow those ions, because as those ions move into the blood, they're going to lower its water potential. Then they're going to move out of the blood into the epithelial cells, lowering the water potential there, which means water's going to go, let's follow down a water potential gradient. The ions go first by diffusion, followed by the water by osmosis, and the end result is water going into your lumen and causing diarrhea and dehydration. And the consequences of diarrhea and dehydration is obviously that it can lead to death, especially in young children. Lots of people, about 100,000 people die every year from this disease. So that's a lot of information, but here's why I reckon you should learn this, okay? I'm going to show you how it can be applied next. So this is why I reckon you should learn this. So once again, speeding through the explanation. I've said it about too many times, but yeah. Binomial name, Vibrio cholera. Cholera is a bacteria, or bacterium if you're talking singular. It has all the same things as other bacteria, so it's a prokaryote, so it's got the DNA loose in the cytoplasm, it's got the plasmids, it's got the slime layer, the capsule, it's got the peptidoglycan in its cell wall or murion, it's got the flagellum, but the unique thing that's beyond your spec is the flagellum is a corkscrew shape, allows the cholera to burrow through the mucus layer on the outside of the epithelial cells, which I've showed here. Once the cholera is inside the epithelial cells, it's producing a toxic protein. And I didn't mention that it's a toxic protein. So when you said, what are the proteins in step one? Just call it a toxic protein. Okay. And it binds to a carbohydrate specific receptor on the outside. That's what allows the cholera to affect or infect epithelial cells in the small intestine. Those receptors are only found in the small intestine, so cholera can't affect your lungs, it can't infect your heart, it can't infect your liver. The second toxic protein binds to chloride ion channels, opening them, allowing chloride ions to move into the lumen, and that lowers the water potential in the lumen, causing water to move out of the epithelial cells into the lumen, therefore lowering the water potential in the epithelial cells, but also setting up an ion gradient, a diffusion gradient of ions from the blood and the body cells into the epithelial cells, which continually causes water to then follow from the body cells and the blood into the epithelial cells and into the lumen by osmosis down a water potential gradient, meaning that you get diarrhea and dehydration, which can lead to death. So that's your kind of extracurricular beyond the spec knowledge. Now, how do you apply this? Why should you bother spending maybe 15-20 minutes learning that? Okay, I'd say the thing I haven't written there is that it produces a toxic protein. So I'm just going to add that on here. Cholera produces a toxic protein or proteins, and the toxic protein binds to your carbohydrate specific receptors on the small intestine, and it binds to the chloride ion channels, opening those as well. Okay, so it has two effects. Now, how can we apply it? Okay, so let's have a look at these essay titles. You can actually fit, once you know that off by heart, so spend 20 minutes learning that off by heart, it really covers half of the essays they've ever asked in the last 20 years. You can apply that one bit of cholera information to half, 50% of the essays. So there's a good chance you're going to get an essay on Wednesday that you can apply the cholera to. But I'm going to do a live later with some other extracurricular bits. So how would you apply it to the importance of ions? So this is an essay that's come up 3 times in the last 20 years. Anything about ions and the usefulness of ions. Well, we're going to work in the whole explanation. So you just have to learn that whole explanation. But your link to the importance of ions, you can talk about the chloride ions. Importance doesn't mean that it's got to be positive. It could be negative, a negative impact. So ions can negatively impact things as well. So cholera, and then you would causes chloride ion channels to open. So there's your link, the effect it has on the chloride ion channels. Okay, let's look at this next one. How are we going to link it if we get any question on proteins? The toxic protein, remember? So it produces a toxic protein. And so that's how I'm going to link it, and that's my link in to proteins. That toxic protein, remember, has a unique primary structure, which gives it a unique tertiary structure. There's my A level, which allows it to bind specifically to those carbohydrate receptors on the epithelial cells of the small intestine. Does that mean I can explain the rest of the cholera stuff? Yes, you'll have time to write a paragraph, a small paragraph, just explaining how cholera makes people ill and stuff, and then saying, well, look, because of this toxic protein with a specific 3D shape, you can mention primary and tertiary structure, that it can bind to the specific receptors that are found, and receptors are made of protein too, that are found on the epithelial cells of the small intestine. Oh, look at my spelling for cells. Jeez, man. Yeah. So there's your link there. What's the importance of osmosis? How can we link osmosis into the cholera data? If you get any question on osmosis, how can we link that to this beyond the spec cholera? What do you reckon? How can I work osmosis in there? Well, it's really so remember what we want to say is the water potential, you're right, increases in the epithelial cells. That's right, which means water moves by osmosis into the lumen, creating the diarrhea. So we're really saying that's where it links. So brilliant. Increase WP in epithelial cells. But actually, remember, it also decreases... Where's my downward arrow? I can't find the opposite on the keyboard. Why don't they put that? You think they'd put that right next to each other? All right, let me get rid of that. Right. Anyway, let's just say how it affects water potential in lumen and epithelial cells. The problem when you do a live, you suddenly remember something that you forgot to mention. So the first step, when the chloride ions move into the lumen, they lower the water potential, which I think you mentioned, Betty, earlier. They lower the water potential in the lumen, causing water to move into the lumen. They also are going to increase the water potential in the epithelial cells with the absence of chloride ions. That's going to affect the water potential. So they will increase the water potential in the epithelial cells, but the main thing is they're decreasing the water potential, obviously, in the lumen as well. So it's not just one going up, but one set of cells is going up while the lumen's water potential is going down, creating that gradient. So epithelial cells losing chloride ions, water potential going up. Lumen is gaining chloride ions and its water potential going down, setting up that water potential gradient for osmosis. So we can talk about that and link it there. And of course, what's the importance of that to living organisms when you've got to link that to the question? Well, dehydration, diarrhea causes dehydration, and all the things we need water for, hydrolysis, all these things, it's a solvent, all of that. If you don't have enough water in your body, you're going to overheat, you're going to die basically, and that's what happens. 100,000 people dying a year. How can bacteria affect the lives of humans and other organisms? Well, we've just you can really just bust out the whole explanation on cholera, and it's going to kill hundreds of thousands of humans every year. And cholera is spread through dirty water and fecal matter. So if you get one on diseases and how they spread, or relationships between organisms, well, fecal matter is like poo basically. And when sewage gets into streams and rivers that people are drinking from, because that's the only water source. Fecal matter, the science word for poo, the cholera is in the fecal matter and the sewage, and that is how people drinking dirty water is how you get infected with cholera. And then once people are have got diarrhea, it's spreading even more. So we can also talk about relationships there between organisms. So if you get any question on relationships between organisms, we can talk about cholera's effect on the human's intestines. Different types of relationships, interactions between organisms, same thing. So this is not a positive symbiotic relationship. Got some more essays where we can talk about this. As I say, I'm only jumping on for 10 minutes here, like 10-15 minutes. I'm going to come on later with some more information that can go to other essays. So importance of shapes. So in the chat, what has cholera got to do with shapes fitting together in cells and organisms? What has shapes? Shapes in biology. How does that link with cholera? And receptors, how does that link with cholera? And movement, how does that link with cholera? Yeah, love it. And those carboreceptors are found, yeah. So, and it's not the cholera that's complementary, remember. It's the toxic protein produced by cholera. Really good word, complementary to receptors on epithelial cells. Nice. Yeah, and that pretty much covers. Whenever I spell receptors, well, type it, not spell it. I missed that out. Yeah. Yeah, but that's cool. That's cool. It's just get that technical language right. So, yeah, it's the toxic protein released by the cholera is complementary to the receptors on the epithelial cells. And you can work that in. And the importance of receptors, well, it means because the receptors are different, cholera only affects so cholera is only affects epithelial cells in SI. It can't bind to receptors outside your heart. It can't bind to the receptor cells on your pancreas or your liver or any other organ. So it can only, I think tonight I'm going to do a live at 8. I don't know whether to just go over, I'm not sure what would be more useful. I'm going to go over some other information that could be extra for essays. I could go through how to plan some essays. Water regulation, obviously cholera fits in there as well, guys. That's pretty obvious. It's going to disrupt your regulation of water because you're just diarrhea, you're losing all the water. And then the diffusion, that is the concentration gradient set up by the removal or the opening of the chloride ion channels. So I'm going to shoot off in a second. Diffusion gradient set up by opening of chloride ion channels. And why is that important? Well, it's that diffusion gradient of the that's set up with the chloride ions, so they keep moving from the cells to the blood to the epithelial cells to the lumen. That leads to water potential gradient. Yeah. Betty, what do you want? What should I go over in my live tonight at 8? What do you think? What sort of questions? Should I just go over essays or more extra content? There's other extra content for like energy transfers. You can't really use cholera massively in energy transfer. There's biodiversity, kind of ecology questions. It's not so easy to work cholera into. Obviously, proteins, ions, water, diffusion, osmosis, it's right there for all of those. Shapes of molecules and receptors. Cell membranes, I haven't put one here, but I know that certain people YouTubers have been saying cell membranes is a big chance of coming up. Obviously, this has a massive effect on cell membranes. Yeah, I'll do some more on the extracurricular because I think if I cover all the other ones, which is the ecology ones, and there's a few things that you can do which cover all the different ecology ones as well. But I would say this is going to cover half of them. This is going to cover proteins. This is going to cover receptors, shapes of molecules, diffusion and osmosis, so the movement in organisms and between organisms, those kind of questions. Water regulation and the importance of water, this is going to cover. You can work it in, but you've always got to link it back to the main question and say, oh, you know, water's so important because of this. But if you have cholera, explain what cholera does and the mechanism, because that's the A-level stuff. And then you're going to lose all this water and it's going to cause dehydration, and you won't be able to do all these other things that water does, like cooling you down, high heat of vaporization, all of that thing. That it's a good solvent. You won't have water to do all that stuff, hence why it kills you. The importance of diffusion as well, that will cover. Cool. All right, let's leave this here. So what you need to do, learn all this info about cholera, and then have a look back through. And these are just some examples I could think of. These are 10 essays that were asked over the last 15 years. They're not the full name of the essay title. I've just taken little bits of them out. So it's not the full essay title. It's just parts of the essay titles from AQA. But all of these are legitimate ones that have come up. So microbes, you can work cholera in there. The importance of microbes, bacteria, or microorganisms. Cool. All right. Nice one. Take care. I'm shooting off. So, no problems. Yeah, I'm going to come on at 8 p.m. and I'll go over some more. I might even, yeah, I don't think I'll come on before that. But I'm going to come on at 8 p.m. and then go through some more. And then I'm going to do a couple on Tuesday as well. All right, take care. Thanks, Betty.